In this review video, we're going to look at the activity of neurons and talk a little bit about some of the structures that are involved with their activity. So let's go. So neurons in the nervous system, we're interested in looking at these key concepts, that neurons and glial cells really make up our nervous system, and that neurons play the starring role because of how they're able to generate communicate and generate these electrical signals, which allow for further communication with other cells and other neurons at those places we call synapses. So why do we care about nervous systems, and why is it so important that ours has evolved as to be as complex as it is? Well, it's been very helpful in our survival. Most animals have some form of a nervous system, some very primitive, where it's simply a net of nerves around their body, but we have developed this very elaborate system which really functionally helps us in a million different ways. Our nervous system is able to communicate very, very rapidly with different places in the body. This is why animals primarily need a nervous system. They need some way of communicating between what they see, so in their visual world or hear, and how their body responds to that. So neurons can move signals at a very, very rapid pace, 20 to 100 meters per second, so pretty impressive. We know that the nervous system has both neurons and these supporting cells called glial cells, sometimes called neuroglia, and you can see some of them here. We have the microglia, if you remember what they are, and there's the oligodendrocytes. There's an endothelial cell, which we have looked at in terms of, we call it also an ependymal cell, but we'll talk about those in a minute. But the idea is that in most cases we can see that it's the neurons that are the ones that are sending the electrical signals, and the rest of these supporting cells, these glial cells, are doing something to support that activity. So if we look at just your basic neuron, we know that there's these four areas, dendrites, that are found on the cell body, and notice how you can see these can be very long, and we talked about that yesterday. The cell body itself, which is a cell, right? It's what all cells have. It's got all the things it needs to make proteins, and to which is critical, so it'll have, and to get energy, so we have mitochondria and all those things that any cell would have. We have an axon. This makes them very unique. Neurons have these long processes we call axons, and then the presynaptic areas, the terminal end here, these little swellings, sometimes called boutons, are what is sort of finalizes this neuron. So then we see another neuron following it. They're very adapted. You know this. We've spent a long time talking about this. Neurons, in general, have this ability to create an electrical impulse by shifting the ions from the inside to the outside of it. So creating a current that runs along the length of the axon, and then once it's hit the end, it's going to release some type of chemical, neurotransmitter, and then it's going to set up this stage for the next neuron to do the same, or to do something else, depends on the neurotransmitter. So that synapse is that little gap. When we say a neuron innervates, the term innervate, it has to do with where which direction it's sort of going and contacting. So in this case, when we look at the axon terminal, in we see that this neuron, we're looking at motor neurons now, this neuron is activating some smooth muscle. Could be some organ that you have. So it's innervating it. Over here, it's also innervating it, so they're just showing, in this case, we have some other intermediate cell or neuron here. So we have some different ways to look at this, but mostly what we're saying here is if we're looking at a bunch of neurons activating some muscle, and we're looking at it in the peripheral nervous system, because we're talking about muscles, Then we can call the bundle of these would be called nerves. So when we say I have a nerve problem, that's what we're really referring to in most cases. In the brain and the spinal cord, however, we call those same long extensions or bundles of neurons that are sort of acting together, we call them tracts. Here's some examples of neuroglia. We have our astroglia or astrocytes, microglia. Here's the oligodendro, they call it glia here, but or oligodendrocytes. So all of these form different functions to connect with the blood vessels and create a bridge between our body and our blood system. So blood-brain barrier, to kind of clean up areas around the synapse, etc. So regulation. It's all about how do we support the neurons. So here's some of the roles of these neuroglia. We've mentioned this already, what astrocytes do to form this blood-brain barrier. Oligodendrocytes are the ones that create the myelin in the central nervous system. And then the Schwann cells are the same kind of, they're the same type of cell in the peripheral nervous system. And they too form the myelin around neurons in the peripheral nervous system. So those are Schwann cells. Here you can see an example of it. So the cell itself is the purple, the darker purple. It's wrapping around like a jelly roll, and here's the actual axon on the inside. Here's the nucleus of that actual Schwann cell, and you can see so in the peripheral nervous system, where the Schwann cells individually wrap, like here's one and here's another, and we could just keep going down. All of those are individual Schwann cells. But in the central nervous system, we're looking at the astrocytes, when we're looking at the oligodendrocytes, you could see that one oligodendrocyte was reaching out and wrapping in several locations. So neurons generate these electrical signals, and they do it, as you know, because they create this gradient of a lot of sodium ions outside, and more of a negative charge is maintained inside a neuron, and that difference in the electrical potential, right? So we talk about potentials a lot, is really what can be referred to as voltage. We can determine the difference in voltage from the outside of a neuron to the inside, and that is what we typically talk about when we're looking at resting potential as being that negative 70 millivolts, that difference in electrical potential on the inside of a neuron versus the outside. Once we allow those voltages, once we allow those difference in voltages to do something, for the ions to move, then we create a current. So it might sound familiar with some of the physics stuff you did quite a few years ago. This I liked only because it tells you a little bit about the concentration gradients outside the cell of sodium, potassium, chloride, and calcium. Few negative charged proteins outside, but then on the inside, very few sodium relative to the outside, a lot of potassium relative to the outside, few chloride again relative to the outside, almost no calcium. Calcium is in fairly small quantity outside, but then the proteins, you can see there's many negatively charged proteins on the inside of a neuron, so the intracellular side. All right, so we're looking here at the resting potential, and just looking at how they measure that, and we've talked about this a little. You can see this, pause it and read it if you'd like. Moving on, though, we know that all of this is maintained through the sodium-potassium pump. So the cell is using ATP, is using energy constantly to maintain this gradient of more sodium outside, more potassium inside, because those gradients are constantly changing. Sodium's wanting to come in, potassium wants to go out. There's some ability for potassium in particular to leak out, and so that those gradients have to constantly get reestablished by this pump. This is the whole idea on the potassium channels. We know that potassium wants to leak out through those leak channels, but there's a point as potassium's leaking out that it's going to be kind of pushed back a little bit by what happens on the outside. So in other words, the balance of the two effects. So we have a concentration effect, how much of that sodium or potassium, in this case, is on one side of the membrane or another, knowing that that usually means that wherever it's in high concentration, high potassium, it's going to want to go out to where there's less of it, or a low concentration. However, the other thing that happens is that we also have inside the neuron, right, we have these negative charged proteins that kind of are always there. Not kind of, they are, so they can't leave. And so they're attracting potassium in some way. So while potassium wants to leak out due to its concentration, it also has there's an attractive force from the negative charges here, and kind of at some point, too many positive charges out here would sort of repel more potassium from coming out. So this is that equilibrium, electrochemical equilibrium for potassium when we are looking at those two things that are working sort of in tandem. This is looking at the different receptors. We talk about these a lot. Mostly we're going to be talking about in the early part of an action potential, early part of activity of a neuron. We look at what's happening on the dendritic side of things, right? So we're going to look at whether or not we're looking at ionotropic receptors or we're looking at metabotropic receptors. So we'll talk more about these. Hopefully these are very familiar to you. We know that there are two different types, that some neurotransmitters operate with one or the other. Some have both types of receptors. And we'll go on from there. So on the ligand-gated channels are the ones that we see on the dendritic spines. These are the ones that are going to open in response to some neurotransmitter landing on them. And the neurotransmitter stays outside in the synapse, but the ligand-gated channels are going to open and let some ion in. Okay, so most of these we would call ionotropic. They're going to open and close relatively quickly, and they're closed when the neuron is at rest. Most of them. Voltage-gated channels require a shift in the potential of the neuron. So we look at what's happening when there are some ions that come in, and it has to reach a threshold in order to open. So just to show you here, so the ligand in this case would be the neurotransmitter. It's going to bind here, and in its binding it's going to basically change the shape of this protein that's at this channel, and that opens it up and allows for some ion to go through. The other, so that's, let's see if I got that one. Oh yeah, here's the voltage-gated channels. So these are closed and only open when there's a shift in the charge of the membrane. There's another one, but we really haven't talked about stretch or tension-gated channels, so I'm going to leave that one as it is and stop right here, and we'll take up with the action potential.